My name is Luke Judge. I'm a physician scientist and a postdoctoral researcher at UCSF and the Gladstone Institute of Cardiovascular Disease. And we're in the laboratory of Dr. Bruce Conklin. My research is focused on understanding the mechanisms by which different genetic mutations lead to disease, either protecting against disease or causing disease. The diseases that I'm mostly focused on are diseases of the muscle, both the skeletal muscle and the heart muscle, and we use human stem cells to study this process. I grew up in Moscow, Idaho, which is a small college town in North Idaho. My dad was a lawyer and my mom was a university teacher, but I was always interested in science. Even as a child, I was interested in tinkering with things and doing experiments. The research we're doing really has the potential to change the way that we study human diseases, because by using actual human cells and human tissues that we can manipulate in the laboratory and do things that we were never able to do before, and access human tissues that were never able to be accessed before. I take care of children who have genetic diseases early in childhood. Most of the severe genetic diseases manifest early in life, and some of those diseases that affect the heart and the skeletal muscle are the ones that really don't have any cures or even good treatments for. I think that part of the problem is that the heart muscle doesn't regenerate itself. So we think that mostly you're born with all the heart muscle cells that you're going to have, and that has to last you for your entire life. And also the heart is a unique organ because it's constantly working, beating 60 to 100 times per minute for your entire life, circulating all of your blood volume throughout your body. And so it's under sort of tremendous mechanical forces, mechanical work, stresses that are really unique to other organs in the body. A typical day is culturing human iPS cells that are maintained and then also differentiated into beating cardiac cells in a tissue culture dish, creating new genetic vectors for manipulating our cell lines, and then interacting with a lot of the other scientists, both in our lab and in the labs on our floor and in other institutions as well. So it looks like this experiment actually worked quite nicely. I can see we have a nice mutant population from the assay there. Collaborating is really satisfying because everybody has different areas of expertise, different backgrounds, different training that they've had, and everyone always thinks of ideas that you wouldn't have thought of yourself, but oftentimes those discussions can lead you to kind of have the critical insight you need to push your project farther. I chose UCSF for a lot of reasons. One, just being the great reputation it has, the great location, the wonderful city it is to live in, but also just scientifically because it has been sort of one of the leaders in the field of regenerative medicine and stem cell biology, which I think are going to be really important tools for treating the diseases of the heart and the muscle. And the Conklin Laboratory is, in a really great environment here at the Gladstone, with a lot of labs who are developing the cutting edge techniques that are needed to develop those kinds of treatments. I think we'll really be able to better understand and investigate all kinds of human diseases, and be able to make predictions that are going to be more reliable for developing the drugs that are going to be effective in treating those in the future.